Welcome back, everyone. Ready for another deep dive? Today, we're getting up close and personal with bacteria.
Specifically, how we can identify these often invisible organisms.
And we're doing it with a test that sounds a lot more complex than it actually is, the Indole Test.
Which lets us differentiate between different types of bacteria.
Like having a microscopic fingerprint kit.
That's basic.
So, where do we even begin with a name like Indole Test? Well, it all comes down to a bacteria's ability or inability to produce a very particular enzyme, tryptophanase.
Now, before anyone runs for the hills at the word enzyme, trust me, this is fascinating stuff. It all starts with an essential amino acid, tryptophan.
Right, an amino acid, those building blocks of proteins we always hear about.
Exactly. And proteins, as we know, are essential for all living things.
Including bacteria.
Exactly. So some bacteria have this neat trick. They use an enzyme, tryptophanase, to break down tryptophan.
A metabolic process that provides them with energy.
And like any good energy conversion, you have byproducts.
And that's where our Indole test comes in, right?
You've got it. This process leaves behind a very specific clue.
A clue in the form of a substance called, you guessed it, indole.
And the Indole test, well, it's all about detecting that telltale indole.
Revealing whether a particular bacterium has the superpower, if you will, of producing that tryptophanase enzyme.
Think of it like a bacterial talent show, and we're looking for the ones who can break down tryptophan.
Okay, I like that analogy. So how do we take this from theory to practice? Imagine a scientist in the lab, they've got their bacterial sample.
First things first, we need to give those bacteria a good environment to grow in.
Set the stage for them to do what bacteria do best.
Multiply, and that happens in what's called tryptone broth, which is, importantly, packed with tryptophan.
Their food of choice, or at least one of them, if they have that special enzyme.
Precisely.
And they hang out in this broth for a while.
About 24 to 48 hours, giving them time to grow and do their thing.
Set the stage for the big reveal.
Exactly.
So the bacteria have had their feast. They've been multiplying in the tryptophan-rich broth.
And now, the real detective work begins.
We introduce a special guest to the party.
Kovac's reagent.
Now, this reagent sounds pretty official. What's it all about?
It's our indicator, designed to react with any indole that might be present.
So it's like a chemical litmus test.
That's a great way to put it.
And what does it look like if we have a positive result?
You'll know it when you see it. A vibrant red ring forming at the top of the broth.
So if our bacteria have been busy breaking down that tryptophan, producing indole, the Kovac's reagent swoops in and creates this red ring.
Precisely. It's the visual confirmation we need. A red ring. They've got the tryptophanase enzyme.
But if the broth remains unchanged, no red ring, we know those particular bacteria haven't been producing enough indole.
At least not enough to react with the reagent. They likely don't have the enzyme, or at least not in significant quantities.
So it's like a bacterial fingerprint analysis.
I like that. We're looking for a match, a chemical signature that tells us who's who in the bacterial world.
And this matters because
it lets us quickly and effectively differentiate between various types of bacteria.
Which is crucial for a whole host of reasons, right? Diagnosing infections, tracking disease outbreaks.
Absolutely. Knowing exactly which bacteria we're dealing with can make all the difference.
So give us an example. Let's say we're looking at Escherichia coli.
E. coli, a classic.
Often linked to food poisoning, though I know it can be a harmless resident in our guts, too.
That's right. Now, E. coli consistently aces the Indole test.
Right.
Red ring. It produces tryptophanase.
So our scientist sees that red ring. They can say with reasonable certainty they might be looking at E. coli.
Among other possibilities, of course.
Okay.
It helps to narrow things down.
Makes sense. What about a bacterium that doesn't produce a lot of tryptophanase?
Well, let's take Enterobacter aerogenes, another common one.
And how does it fare on the Indole test?
It usually flunks.
No red ring.
Exactly.
Yeah.
So right there, with this one test, we can start to differentiate between these two bacteria.
And that can be absolutely critical in a diagnostic scenario. Knowing who the culprit is allows us to target treatment much more effectively. It's amazing how much information we can get from one little test tube, right?
It really is. The Indole test gives us a great starting point. By understanding which bacteria produce tryptophanase, and therefore test positive, we can start to narrow down the possibilities.
And that's key, especially when time is of the essence. But it sounds like the implications go way beyond just identification.
Absolutely. This knowledge allows us to study these microorganisms in much greater depth.
So we're talking about understanding their metabolic pathways, how they function in different environments.
Exactly. Even their potential for applications in biotechnology, which is a whole other fascinating area.
It makes you wonder, could we target this enzyme, tryptophanase, in developing new drugs or treatments?
Now, that's a fascinating question, and it's a possibility researchers are definitely exploring. Knowing how bacteria use something like tryptophan could offer new targets for antibiotics or other therapies.
It's incredible to think about how much we can learn from something as seemingly simple as a color change in a test tube.
It just goes to show the power of scientific inquiry. We start with the question, can these bacteria produce this enzyme? And it opens up a world of new knowledge.
A deeper understanding of bacteria, their metabolic secrets, and even potential avenues for future research.
A perfect example of how a deep dive into a single topic can unlock a universe of possibilities.
And on that note, we've reached the end of our exploration for today. But the beauty of science is that there's always more to discover. Until next time, keep those minds curious and remember, sometimes the most fascinating journeys begin with the simplest of questions.